This is the walkthrough video for Wednesday. It is February 26th. Ooh, that kind of sums it up. Hey, we're going to do a practice problem about like unicorns, just so there's like no connection to animals you may or may not know. So, this is one of those problems like the one yesterday about patients, humans, right? And it gives you that recessive allele information. And so the question is asking you if you know the percentage of a population's unicorn that have the recessive phenotype, then you need to figure out from this, are you looking at p squared, 2pq, or q squared? So just a reminder that these two together in a simple dominance and recessive situation give you the percentage that has that dominant phenotype. And then this is the one that gives you that recessive phenotype. It's the percentage of homozygous recessive, right? All of these are genotype percentages. These are the ones that are allele percentages. So I've drawn a little picture here to help you think about it. If you know q squared, first you have to know that they're giving you q squared, but I'm telling you they are. So if you know q squared, you can take the square root and find q. If you can find q, you can find p. If you can find p, you can find the other things that are in this equation. So hopefully that helps you. If it's still hard, come see me. I will try to take care of you. All right, so we're still thinking about whether populations are undergoing genetic change. And this time, we're going to begin to think about using real data from rock pocket mouse populations as a real life example of what it looks like to do this modification and what happens as a result, right? So if you were here yesterday, you saw this charismatic video. If not, you can go up in the slides and watch it about these Snicker bars of the desert. And today, we're going to think about what it looks like to, oh, you can't see that at all. Please hold. Please hold. Oh, why is it so hard? Stop, stop. Oh my gosh. How you doing, Dr. Milks? I can like not see straight right now. There we go. Cool. So this is the assignment you're going to take care of. It is a story about these really cute mice, like we talked about. They're like shorter than an average pencil from nose to tail, and they have just a handful of paper clips worth of mass. And they've been so important. So you've watched the short film, you're able to read A and B. You might need to look up C if you've forgotten, but I will actually tell you actually because I'm feeling generous. It's the MC1R gene, which is or other proteins in that pathway that end up making melanin in fur. So fur color is the specific trait and has to do with cell signaling that turns eumelanin pigment production way up in those mice. So you watch the video. This is a tutorial. You might need it if you haven't watched and taken notes on the 7.5 video, or you've missed class earlier this week, or you just want a quick refresher. And I think the thing that you probably need to start with is that we are going to be working with two alleles at one gene, a dominant and a recessive one. And then you're going to want to start kind of here. And that's where the assignment's really going to pick up for you. So you're going to see a sample problem very similar to the one that you found, except you have to calculate it q squared. So this time we're telling you there are 100 mice and that 81 individuals have this color and the other ones either have the other genotypes. And so you have to do some division as kind of described here. So we're going to have you do some Hardy-Weinberg calculations. You'll see them here. And then you're going to apply those to real life Nachman lab data across the field where they are collecting them, these dark rocky lava outcrops and these light rocky granite areas. And so you'll be doing some calculations. Be really careful about noting which one you are calculating. And then you're going to begin to like think about using information like this to back up some claims. Most of you are not going to get to part 3, I don't think, until Thursday, which is fine. And you're going to use an equation spreadsheet, which is given for you in Canvas today if you get this far, to do a little of this analysis. And you can work through this. But this is where we're going to start our tutorial tomorrow. So I'm going to hang on. I have to share everything again. Oh, it's really difficult. Which is even the thing I'm sharing? Is it this one? Hey, it is. Great. All right. So again, the 7.5 video is where you're going to find it. If you're playing along at home, here is a real life test question. I think you've seen it before, but it's also excellent review. See if you can find the right answer and write about why it's correct. If you can't, see if you can knock out two incorrect answers and tell me why they're wrong. And of course, if you want to go over this problem, you can look in your review book for Unit 2, or you can look in your study folder for Unit 2. I bet you will find it in either of those places. All right, that's it for me. Hope you're doing well out there. Take care. See you soon.